My poster at ARUK 2021 really looks into the relationship between MRI measures of microstructure in the superficial white matter in a cohort of healthy controls, but also young onset Alzheimer's disease. So for example, my research actually is focused on the superficial white matter, which is just a region of white matter that's relatively overlooked in the brain. It's just below the cortical grey matter. Some of our previous results using a type of MRI called diffusion MRI that's sensitive to the direction of water in the brain, it suggests that there's actually a reduction of myelinated fibers in the superficial white matter. But we wanted to really look into what's going on there on the microstructural level as well, using an independent sequence. We used for another method called multi-parametric mapping, and that's sensitive to these different microstructural properties as well. So these participants both had a diffusion MRI scan and also scans with multi-parametric mapping, and then we essentially overlaid these metrics and correlated them, looked at the correlation between them in different regions of interest. The main takeaway is that the measures in the diffusion MRI method that relate to myelin, for example, are related to the measures in the multi-parametric mapping technique that are also related to myelin, which suggests that they are picking up on the similar underlying microstructural properties. Also, in terms of looking not just within the superficial white matter, but in the surrounding tissue. So when you think about going from the cortical grey matter down into the superficial white matter, they show similar trajectories as well, which suggests that there's a similar, picking up on similar microstructural properties in the surrounding tissue. So really this kind of lends credence or corroborates our inference of our previous findings that there are microstructural changes going on in this region, particularly myelin, but also in other microstructural elements as well, for example, in iron, which has been recently suggested could be a marker for this superficial white matter. So this is it's actually very preliminary work at the moment. It's really a first look into these relationships. So we're currently looking at this in a more exploratory way, instead of just regions of interest, looking across the whole brain, while also controlling for other covariates like age and sex and things like cortical thickness that could affect some of the measures. And another thing I should really mention is that a main limitation of this work is the spatial resolution of our images. So, for example, the metrics that we've extracted could be influenced by surrounding tissue. And we're currently working with some amazing collaborators to look at this in much higher resolution images to see how specific this is to the superficial white matter tissue. I think this is really going to help understand what's going on the microstructural level in people who are living with the disease. Really, I think the kind of the story of superficial white matter is relatively untold in neuroimaging, in neuroanatomy, and especially Alzheimer's disease. The few studies that have looked into superficial white matter, there are methodological limitations associated with that. So this is really helping drill down into what could be going on there. And if we can extract more meaningful information from MRI scans, it could help us put people into certain disease stages, or potentially see changes that could occur before symptoms. But that's still early days at the moment. I think we're still looking at the kind of what's going on the microstructural level before we can really apply it to that. But it's really about extracting meaningful information that could help provide more information on top of things like cortical thickness and atrophy and things like that.